Hello, we're glad you've joined us for this live webinar, Practical Considerations in the Design of Bottom-Up Mass Spectrometry-Based Proteomics Methods. I'm Judy O'Rourke-Laberts, and I'll be moderating this session. Today's educational web seminar is presented by LabRoots, the leading scientific social networking website and provider of virtual events and webinars advancing scientific collaboration, and is made possible by an open educational grant from Agilent. Agilent has had no input in the selection of speakers, content, or mode of presentation. So let's get started. You can pose questions to the speaker during the presentation while they're fresh in your mind. To do so, simply type them into the drop-down box located on the far left of your screen labeled Ask a Question and click on the Send button. Questions will be answered after the presentation. To enlarge the slide window, click on the arrows at the top right-hand corner of the presentation window. If you experience technical problems seeing or hearing the presentation, just click on the Support tab found at the top right of the presentation window, or report your problem by typing it into the Answer a Question box located on the far left of your screen. This is an educational webinar and thus offers free continuing education credits. Please click on the Continuing Education Credits tab located at the top right of the presentation window and follow the process of obtaining your credits. I now present today's speaker, Sandra E. Spencer, Ph.D., the Proteomics Platform Manager in the Morin Lab at the BC Cancer Research Center. Dr. Spencer's undergraduate degrees are in chemistry and biochemistry. At that time, she did research with Dr. Kevin Schug at the University of Texas at Arlington, investigating the toxic components secreted by the fish-killing algae Promnesium parvum. She did graduate work in aerosol chemistry and mass spectrometry instrumentation under the supervision of Dr. Gary Glish at the University of North Carolina at Chapel Hill. Her thesis was on real-time analysis of aerosol particle composition using novel ambient ionization and gas phase separation techniques. Dr. Spencer did her postdoctoral research in the MacCoss laboratory at the University of Washington. There, she primarily worked on method development for targeted proteomics and improvement of nano-liquid chromatography robustness by implementing robotic solutions. Dr. Spencer will now begin her presentation.
Thank you for that wonderful introduction. So most of this work was actually performed at the University of Washington during my postdoc, which is why it's all on Washington UW background, even though I am currently in Vancouver at the BC Cancer Agency. So I'd like to start this out by asking and thinking about why we even want to do proteomics. So if you think about the central dogma of biology, we start with DNA, and that's transcribed into mRNA, which is translated into proteins. And if we think about what each of these things do, the DNA is basically the ultimate potential of the cell. It's what's possible, what you could make. Whereas the mRNA does tell us something about what the cell is trying to do, what the cell wants to do. But proteins are what makes it happen. They're really the functional capability of a cell. And I mean, they are harder to measure. We're pretty good at measuring mRNA, right? And so you may ask, well, why not just measure mRNA and infer protein concentration from that? So if we did that, if we measured the mRNA abundance, the probability of observing a given protein based on its mRNA abundance does increase, as shown in this graph. However, if we're trying to predict the abundance of a protein based on the abundance of mRNA, that's actually not so straightforward. So if we look at this correlation, there's an R squared of between about 0.3, 0.6. There are lots of studies that have shown this. And just to exemplify this, let's take one little slice of this data. Look at the mRNA abundance of log 10 molecules, or log 10 molecules per cell. And if we look at that, we can have anywhere from 100 to 10 to the 8th molecules per cell of the protein. Okay, so that's 6 orders of magnitude difference. And so we can't really predict based on the mRNA abundance, the protein abundance. We're very bad at that. So we want to do proteomics. We want to measure these proteins, and perhaps we want to measure their post-translational modifications, which regulate the activity of the protein, the localization of the protein, how long the protein lasts. We might want to do this to get a readout of what the DNA is doing. So for example, in Alzheimer's disease, there is a DNA mutation that creates ApoE4 instead of an ApoE3 protein, and that puts you at high risk for Alzheimer's disease. We can look at post-translational modifications as an indicator of the cell status in various signaling pathways. And these proteins also act in disease, and we'll talk a little bit about all of these different things during this presentation. So why is this difficult? Why is it so much harder to measure proteins than RNA? Well, one of the big things is the complexity of the sample, and another big thing is the dynamic concentration range. So let's think about the proteins just in, this is for serum from Anderson et al. In 2002, and what they did was look at the concentration of a ton of proteins in serum. Now note that this y-axis is on a log scale, and there are 10 orders of magnitude from the most abundant protein in plasma to the least abundant protein in plasma, okay? So this is huge, and this is a really difficult analytical challenge. If we think about 10 orders of magnitude, this would be like trying to find a Great Dane between Earth and Pluto, okay? Another thing, because that's really big, it's really hard to imagine how far Pluto is away, right, other than it's really, really far away. So what if we had a little kid painting on the walls, and you take the red pigment out of that paint. You dig into that, get to the iron ion that's making it actually red. And that is 10 orders of magnitude difference in size between that child and the atom. So it's a lot. It's hard to measure all on the same scale. And it's a very complex mixture. Your sample's not just proteins, and different samples have different amounts of proteins and other things in them. Just think about urine and skeletal muscle, say. They have different amounts of water, even though for both of them, yes, water is the most abundant compound. In urine, we're super salty. We have a very saline solution. So we have urea and sodium chloride. And at the very, very bottom of this composition list, we have some other ions, proteins, hormones, metabolites, just a little bit of those. We don't have a lot. Whereas if we look at skeletal muscle, the second most abundant compound is protein, followed closely by liquids and then other things. And so obviously these two samples are going to require two very different methods to analyze the protein, right? So if we thought about this in terms of a haystack, right? These things we want to look at are this haystack. And if we're doing metabolomics, we're trying to find a needle in a haystack, right? Because the needle is so different, right? If I say creatine and urea, those things are very, very different, and I might be able to use chemical methods to enrich for them. However, proteins are all very similar chemically, right? So this is kind of like trying to look for hay in a haystack. If I'm looking for apolipoprotein E in the entire proteome, this is going to be a very difficult challenge, right? Now I want to switch gears a little bit and talk about the two different types of proteomic experiments that you can do, right? We can do discovery experiments, which is asking the questions, what proteins are in my sample? And this tends to be a very, very commonly asked question. What's here? Why is this different? Why is disease A different than healthy? But we can also do a targeted experiment. And there are certain instances when we want to do a targeted experiment. And that's when we want to ask the questions, are proteins A, B, and C in my sample? Okay? Some pros and cons of these two. In the discovery experiment, you don't have to know what you're looking for. You don't have to have a hypothesis about what is there and what's not there. It's supposedly unbiased. There are certain more and less biased discovery techniques out there, but it is slow and it requires a really nice machine, and so it's really expensive. On the other hand, targeted experiments can be very, very fast. They're relatively inexpensive, and you get a higher sensitivity because you're looking exactly at what you want to see, exactly at what you want to know. You're asking a very targeted hypothesis. However, if you don't know to look for it, you're not going to see it ever, right? You're going to have to go back to those samples and analyze them again if you come up with a different hypothesis. But in this talk, my goal or one of my goals, is to convince you that sometimes we need to break up with discovery proteomics, right? It sounds really nice. We're all very scared of not seeing something that might be there. But if you know what you're looking for in a proteome, if you want to run an assay a lot, if you want to reduce your assay cost, you might need to do targeted proteomics, okay? And so we're going to talk from here toward the end about different ways to develop a targeted proteomic method, specifically quantitative targeted proteomic methods, okay? And I'm going to show you how to do this in 2 easy steps. What we want to do is start with this list of targeted proteins and turn it into a refined SRM table, right? So step 1 is that you're going to do some stuff to get a list of peptides that you want to look at. And then you're going to do some mass spectrometry magic to check these peptides out and get your refined SRM table, right? Super simple, right? Well, we'll walk through a bunch of different ways to do it. So the first way we could do this, I like to call it the spray and pray method. So what we're going to do is take one of these proteins that we're interested in. We will highlight all the tryptic peptides. So we're using trypsin usually for digestion just because it's very, very reproducible and predictable, okay? So we'll look at all of our tryptic peptides, and then you go and buy all these tryptic peptides. You pray to the mass spec gods that be that some of them work. You run them, and you get your SRM table based on what worked. So there's a few good things and bad things about this, right? It's expensive, for one, and it uses mass spectrometry. So we like that part, right? We like actually collecting this data to look at it. But it's really expensive, and having had to do this a couple times, it's somewhat infuriating to go through and then you don't know if it's the peptide that's bad or if the instrument's bad or if your column went bad. It's just a little bit time-consuming and tedious, it seems. So we can do better than just guess and check, right? We can predict what we're going to see. We're going to use an educated guess now to get from our list of proteins to our SRM table. So we'll do the same thing. We'll take our protein, we'll digest it in silico, predict what peptides are going to come out, but we're just going to do that on the computer, right? Well, we're just going to use the computer, hope that we narrowed this list down well enough on the computer for things like peptide length, right? Really short peptides don't fly well, really long peptides don't fly well, so we're going to have an intermediate peptide length, a good hydrophobicity for chromatography. We're not going to have a whole lot of amino acids that can get oxidized, and then we'll do the same thing. We'll buy those ones and run them. And this sometimes works. So let me show you this for one protein. So here's a protein sequence, and I used ESP predictor in purple here to predict all of the peptides that might come out of this, right? Now, I just want to note that the highlighted brown portion is the sequence that I actually got. We'll talk about this a little later, but I bought this protein, so I only bought a portion of the sequence, not the entire sequence length. So I have the ESP predictor score here in purple. I measured the peak area by SRM here in gold, and we can see the correlation between the two on this curve. I've actually plotted the correlation. So here we've got the ESP predictor rank, so 1 through 10, which one was the best, next to the SRM rank. And it's pretty much a scatter plot, right? One of the best SRM proteins is in the top 3 for the ESP predictor, right? The other ones are lower, and sure, I might pick these to take a look at. They're not super horrible, but it's just not agreeing super well. Now, I'm coming from a targeted background, and so you might be like, well, Sandy, you're probably just cherry picking here. I promise I'm not. Here's 4 other proteins from the same project where we had the same issue. Maybe 2 proteins are in the top 3 proteins, and if you look down in the bottom right corner, like none of the top proteins are in the top 3 that were predicted by ESP predictor. Now, here are just the correlations for the ESP predictor rank next to the SRM rank. And we can look at some other prediction algorithms because I'm not just bashing ESP predictor here, okay? We can go to Peptide Atlas. It's a great database, a great resource, but and what they do is show you a whole bunch of different algorithms all next to each other. So that's what's here on the right-hand side of this curve is for a set of proteins. You've got scores from a bunch of different algorithms, okay? And if we use these Peptide Atlas scores, so here's the ESP predictor score again that we pulled from Peptide Atlas. And we can use the peptide suitability score, right? This is just a different algorithm that's actually worse than Peptide Atlas for this specific experiment. The peptide sieve score also seems to be much worse than the ESP predictor score. And lastly, the detectability predictor score. And again, these are just 4 different algorithms that you can use that have different ways of predicting what peptides are going to show up the best. And so what we're finding here is that they do okay, but if you really want to develop that assay with great signal where you're getting the best limits of detection, the best sensitivity, this might not be the way to start, right? Picking peptides that aren't giving you the best response. And generally, just remember that this machine doesn't have a brain, right? This machine learning algorithm doesn't actually have a brain. But we do, okay? We can get our own data. And we can think about this and say, well, okay, so we need to get our own data. But these prediction algorithms are fed by data. So why don't they work, right? Why can't we feed data in to these prediction algorithms to get out what we want? And so we can actually look at how this data is collected to answer this question. And so a third method for picking your peptides are using DDA data repositories. Okay, so this is data-dependent acquisition, and it's a very common discovery proteomics technique. And so a lot of these various algorithms use DDA data to train, all right? And so this is what I'm showing here from Peptide Atlas, is that this is the empirical suitability score based on the DDA data. And if we look at this, this looks a lot like those curves or those plots that we were looking at before. And so why doesn't this work? Well, because abundance in DDA data is calculated based on spectrum counts. So now on this curve, you see the SRM peak area here on your left in purple, and the right-hand y-axis are DDA spectrum counts, okay? And these don't seem to correlate very well. And because we're training our algorithms with the DDA data, and that doesn't correlate to SRM data, why would we expect those algorithms to correlate, okay? So in summary, why doesn't DDA data work? Spectrum counts is not the same thing as MS peak area, right? So we can also look at the MS1 peak area too to try and do this. So you've got here now SRM peak area versus MS1 peak area, and it's the same story, right? So spectrum counts, MS1 peak area, they're not going to be the same as your tandem mass spectral peak area. And so, well, why don't we just use SRM data to populate SRM transition lists? So we can do that. There's actually from the same makers of Peptide Atlas, there's SRM Atlas. And this is a repository for SRM data. Okay? And this is great. You can use it. The one issue that you might come across here is that in the repository data, even though it might correlate pretty well with SRM data collected on your instrument versus my instrument versus Phil's instrument, you might be missing some data, right? Because with SRM, again, we're looking at very targeted things. These are the peptides that the researchers that uploaded their data were interested in. And so these are the only peptides there's data for, right? And so if we want to use this VNAP, the second most abundant peptide for some reason, we don't have information on that. So in summary, when we're talking about data repositories and prediction algorithms, I can use these. It's not the same as I should use these. And I just ask that you think carefully about how you are, how you're picking the data, how you're picking your peptides, and ask yourself the question, is this what you want for your experiment? Because this is the very base, fundamental, just structure of your assay. And if it's not done well, you're not going to have as good of an assay as you could, right? So just do it. Just do the experiment, okay? And we'll talk about how to do this experiment to empirically derive your transition list for your experiment, right? Here's my general workflow for you. You produce a recombinant protein standard. You monitor, quote, all tryptic peptide, and we'll talk about why that's in quotes. We'll do a peptide response analysis, an IRT calibration. Through this whole time, we're going to be refining our transition list. And then at the very end, we're going to validate our assay. All right. So let's start talking about how we get recombinant protein standard rather than buying these peptide standards. So you can buy it. So that's an option. It's actually often not all that expensive. Sometimes it is. It depends on the protein. You have to just check it out. It's just case by case. I did want to give you an example of when you might have to rethink buying your protein. So, or maybe rethink your supplier. So here's a data sheet for a protein from an unnamed supplier. And if we look in at the various things they tell us about our recombinant protein, there is a proprietary purification process. Okay, that's not too weird. I would really like it if they said like HPLC UV and showed me some spectra, but this is fine. It's also, yes, recombinant full-length protein with an N-terminal proprietary tag. So they have a predicted molecular weight. They're not going to tell me what their N-terminal tag is. And that's really hard for mass spectrometry, right? If you just have a chunk of the protein, if you have a tag that you don't know is there, your sequences are going to be different, right? And you just need to know that when you're going into it. And so anytime I buy protein from a supplier, I actually insist that they send me what they have written down as the sequence for what would be the sequence for the protein. Because if you look at the 70.07 kilodaltons as the predicted molecular weight for the protein, and we look at the UniProt database, that's supposed to be 45 kilodaltons, right? So there's a huge tag on this. There's basically a small protein attached to our protein. Also, this is $650 for 10 micrograms, which is pretty darn expensive. And if you want to look at the data sheet, it's a broken link. So this raised some flags, and I actually ended up going to another supplier, and we'll check out this other supplier. So it's the same protein. You look at the data sheet, they give you a lovely gel with the full molecular weight range, 14 kilodaltons all the way up to 120, right? Sometimes you'll get these where they'll just like zoom in on this range that they expect it to show up in, which, by the way, note that it's 45, right around where we expect where this protein to show up. But they'll just cut out that slice of it and give you that picture, and you won't know like really the purity, right? On the purity, by the way, they say it's greater than 95%. I look at their SDS page gel. I agree with that. And it's a 6 his tag at the N-terminus. They're very clear. They have the gene encoding MET1 to Gly403, so we know what sequence we're going to get out of there. And they tell us what it's in. Okay, so we're in sodium acetate, sodium chloride, and glycerol. So this is really important because some stuff, such as glycerol, makes it really hard to do like an amino acid analysis. Glycerol is a huge interferent there. And so if we wanted to sequence the protein ourselves or do an amino acid analysis of some sort, we might have some trouble with this guy specifically and might have to figure out a way to get rid of that glycerol. But it's good to know about. Option 2 for getting our proteins is actually just to make it ourselves. And so Thermo actually sells this kit that makes it really, really easy to just express proteins in, I believe, a HeLa lysate. So you just, you can expand, purify, and sequence a cDNA clone. You can often just buy those. You do in vitro transcription translation with this kit. And then you can enrich the protein of interest via glutathione saccharose resin. So we've got this glutathione tag on the end of the cDNA clone, and we just pull that down. And then what we can do is actually spike in a heavy label of one of the peptides in the GST tag, okay? And then we can plot this. So here on this calibration curve, we're looking at the ratio of the light to heavy peptide from the GST tag on the y-axis, and then just the concentration of the peptide on the x-axis. And we can actually calibrate this. We can have a pretty good idea of how much protein we've synthesized. And it's cheap. You can make 25 to 500 femtomoles per IVT experiment. It's less than $20 a protein, which is great. And this is enough protein to last for quite a while. So next, now that we figured out how to get our standard, let's talk about how to pick our peptides, okay? So if this white box is a space that contains all the predicted tryptic peptides from a protein, what we're going to do is look at a subset of these peptides that are a 2+ peptide parent ion that will dissociate into a 1+ product ion. Now, if we have room in our method, right, and by room in our method, I mean if we can look at that many things, because we are limited in how many transitions we can look at in an SRM assay at a single time. So we'll look at all the 2+ peptide to 1+ product ion transitions. Then if we've got time, we can look at some confirmatory B ion transitions. And then if we've still got room in that method, I would go in and look at the 3+ to 1 or 2+ B and Y ion series, okay? And this is just because these 2+ to 1+ Y ion peptides actually tend to show up the best. If you look at a lot of SRM methods that have been developed, and even in your discovery data, oftentimes you see that this Y ion series is the one that really guides you to what this peptide was. So let's do an example. Let's look at a protein and look at the portion of the sequence that I purchased. So I only had a portion of the sequence, but here are the options for the tryptic peptides from that portion of the sequence that I purchased. And then we can plot these. So here's our first chromatogram of the seminar. On the x-axis, you see retention time over the course of the experiment. So for those of you that are familiar with Discovery Proteomics and targeted proteomics, you can see here that this is a 20-minute experiment, whereas a lot of discovery experiments are 2 hours plus, right? So this is much faster than a discovery experiment often is. On the y-axis, we've just got the intensity, and we're just measuring the intensity of each peptide over time. And so I've shown you here the response for each one of the peptides from the protein of interest for us. I can zoom in on this x-axis and just kind of prove to you that the peptides that I say are there are actually there. And then, now that we've seen some peptides, we need to constrain this list because this is going to be huge, okay? So if we look at, say, a specific protein, we want to pick out the peptides that are unique to that protein. So peptides that aren't going to be found in other proteins in the proteome, okay? And what this allows us to do is say that if I see, say here, for example, the LAAP peptide, if I see this peptide, I know that this protein is present, right? We also want to remove any peptides that have an ambiguous elution profile. So this is actually just one peptide. It elutes at 2 different points. I believe this one contains proline, which is often the cause of this. If you have 2 proline isomers in a peptide, you'll get multiple peaks. You also sometimes just get chairs out of these guys. So here's a little chair peak. And this is actually my rotation student Deanna's favorite peak. This is the bat peak. Can happen sometimes as well. Now, when we have these ambiguous peaks, we can counteract it a little bit, right? So this is why I said, look at that 3+ charge state too if you can, because if we look at the 2+ charge state for a specific peptide, it may come out with multiple peaks, and we don't know which is actually the one we should quantify on. But if we look at the 3+ charge state, we can pick the peak that corresponds to the elution of the peptide we expect, right? Because in this 3+ charge state, there's only one peak, and the 2+ and 3+ charge state should be eluting at exactly the same time, because it's just that the peptide elutes, and then we make two different charge states out of it. We can also go through, once we've figured out where all of our peaks are, what our good transitions are, and remove low and noisy transitions. So here in red, you see that, sometimes you measure Y ions that just aren't there. And so you want to delete all of those out, right? So we've started with this big net, and we're narrowing everything down, trying to just kick out anything that's not giving us good information. So once we've done that, we can narrow this down to peptides that respond, right? And then the next thing we want to do is actually look at the stability of these peptides that respond, because we want our peak area to change with concentration, not time, right? So we started doing this in the MacCoss lab, where we actually do this incubation for 72 hours of the peptides in the auto sampler at 4 degrees Celsius. Now, there's a very specific reason we use 72 hours. 72 hours is the amount of time that you need to put a sample in on a Friday and keep your auto sampler running till Monday, so that your tech or grad student or postdoc doesn't have to come in on Sunday and maybe try to skip paying for parking and maybe have to talk their way out of a parking ticket on campus. Not that that ever happened, but we're just going to prevent that from happening. But if we look at this data for peptides from this protein, we actually see two of the peptides switch abundance, right, between the 2 days. And we can see this happening here. So this is just the percent change over 72 hours for all of the peptides in this protein. And I've drawn red lines at plus or minus 10%, right? So we're trying to keep that CV really tight. So these two peptides, of all the peptides we looked at, there are two peptides that we think are staying stable for this experiment. And now the MacCoss lab has done a lot of these experiments, and we've uploaded this to passport.maccosslab.org. It's a protein assay portal stability data, and you can actually just come in here and pick any protein you want, go to an interactive view, and get results from these experiments that we did for you. And we measured all the peptides from the sequence of the protein that we had, and so you can go in there and get the information that you want that we've already collected for you. So we now have talked about peptides that respond, and we've talked about now reliable peptides. We also want to make sure that our peptides are selective, that we're selective for our peptides, that they're not interfered with, right? So if we put something in a different matrix, I can measure something in ammonium bicarbonate or a yeast background, but if I put it in human, there's going to be different peptides there, and we're going to have different interferences. So you always want to take your peptides, measure them in a matrix, make sure to remove any transitions that are interfered with, or assess any matrix suppression of peptide signal. So there's a few ways to do this. You can just spike into native. You can use SILAC. You can use a diverged species. So what I use a lot, or have used a lot, is chicken serum for human plasma. And then for clinical experiments, there's actually an assurance interferences kit that Sun Diagnostics sells. It's got lipids and small molecules and a bunch of stuff that we expect to interfere with proteomics experiments. And I just want to show you that this is a real thing. So if you look here at this peptide in a buffer, so I'm showing the 2 and the 3+ transition state. 3+ transition state kind of stinks, but it shows us which peptide from the 2+, or which peak from the 2+ is actually our peptide. And then we look at this in chicken liver background, because this was from human, and we just totally don't even see it, right? So don't be scared. This might happen to you. You want to leave as many options going in as you can. Transitions also get interfered with, and so here's a different peptide that looks great in buffer, beautiful signal. And then when we go into our matrix, we lose an order of magnitude in signal, and we see that some of our best transitions are very obviously interfered with. So for example, the pink transition here has a nice little tail that it didn't have when we looked at it in the buffer. All right. So the next thing I want to talk about is actually being able to do quantitative analysis on these, because what we have are these Gaussian peaks that we're measuring, and we want to estimate the true peak shape by selecting them. So I had a project where I was looking at 658 transitions, and so that's where these numbers are coming from, right? So if I'm measuring a whole bunch of transitions concurrently, right? Say I only get to come back to the original transition, the first transition in my list, every, I don't know, 7 seconds, okay? Well, then I'm only going to take a few points over the course of a peak, and if I tried to trace a Gaussian distribution to fit this, it just totally doesn't fit, right? But if I can take 10 time points across the peak, this actually fits quite well. And so we always want to try and get 10 time points across the peak, and we have about 20 to 30 second peak widths. So if I was trying to measure all of these transitions all at once, I would have this 7-second cycle. That means 4.6 cycles a peak. And that's not going to work very well. So what we're going to do instead is calibrate our assay to an IRT. Okay, so what this means is basically we take a set of peptides that we know span the range across our retention time, and we measure our samples relative to these. So we can spike them in, spike the retention time calibration mixture in, and measure our peptides relative to that. Let me just show you how nicely this works. So first, there's a whole bunch of different options. They all work fine. And if we have a set of peptides, here on the x-axis I'm showing you the index retention time, and on the y-axis is the measured retention time. And we want them to all fall within 5 minutes, okay? And that, or within a 5-minute window, and that will allow us to have 2.5 second cycle time, right? With this 5-minute window, we're only measuring 250 transitions concurrently, which gives us a 2.5 second cycle time. So if we take our proteins that we measure in buffer and throw them in chicken liver, this is about what it looks like, okay? But if we correct for our retention time calibrators, we can actually get them nice and tight back around our original line in our different matrix, okay? And so this is how you can actually schedule the experiments to be able to only measure a small subset of peptides at any given time, kind of save yourself some time. All right, so for the last little bit of this talk, I want to talk about assay validation. Okay, this is kind of the last spot. We have an assay now, but we have to validate it, or else it's not really worth anything. Okay, if we're talking about clinical assay validation, there's 6 points we want to validate: reproducibility, stability, linearity, LLOQ, interferences, and then like storage stability, right? Or stability under stress. And so what have we done so far? Well, we've looked at the stability, the selectivity, and the accuracy. So let's talk about precision. And in talking about precision, I want to talk about some internal standards, okay, and how important they are. So this was a time course, and this is actually ApoA1 spiked in and measured at different digestion times. And I did replicates because every good analytical chemist likes replicates, right? And here are my error bars. They're huge. They're ridiculously huge. And so if you look into this with the 2 different replicates, replicate 1, something happened at the higher time points, right? We're just getting a lower signal for all of the higher time points, which is what's leading to this peak area change. So what I actually did was normalize this signal to my internal standards, okay? If I have internal standards, I can normalize the signal. So this is the peak area ratio to some stable isotope labeled internal standards of this peptide, and I can restore the signal, right? And I can bring down the CV of this experiment or of these measurements, right? It's always important to include standards. Now, you also might want to include a protein internal standard. So back here we're talking about using a peptide internal standard where I spike it in and just measure to look at instrument performance here, right? Because that's probably what happened is my instrument just wasn't performing well. But you can also use a protein internal standard to assess different digestion conditions and sample preparation issues. So for example, in one experiment, I have both a peptide and a protein internal standard there. And so the standard is ApoA1, and here's a peptide from ApoA1. Now, the peptide shows up just fine, but the protein internal standard did not, okay? Or the peptide from the protein. So these are actually differentially labeled. One is 15N and one is labeled on the C terminus. And so what we can conclude here is that the instrument's working fine because I see my peptide internal standard. But something happened with the digestion. I didn't digest the protein, THLA, out of my protein standard. And so my data is in question, right? And so you can real-time monitor and do QC and QA of your experiments by using internal standards. And I just want to give a little bit of a shout out to the internal standard I used. It's currently sold by CIL as Proteus QC. We helped develop it, and so these are the differentially labeled peptide and protein internal standards. So the peptides are labeled just at the C terminus, and the protein is 15N labeled, and so they show up differently. We mix those together and can add them to a biosample and digest that and analyze it and have all-in-one standard that you can just spike in. So that's been pretty nice. And you actually can look at a lot of different stuff with this. So for example, retention time drifts over time. So from injection 10 out to 400 or 145 on a single column, you get some retention time drifts. You can actually use this to help recalibrate any IRT measurements you might be doing. And you can also use it for normalization. So for example, if you just take the peak area of a peptide, our CV might be pretty high because our peak area might drop over time. So this is injection 29, 90, and 120. But if you normalize these to the internal standard, you can really bring this CV back up. So we can restore our CV. We also want to assess our reproducibility, so the CV of the whole experiment. And this is actually done commonly in what's called a 5x5 assay. So you can take a set of samples and store them and analyze 5 samples on each of 5 different days and get the CVs first intraday. So on each day, you can take a CV of samples from day 1, A through E. Then you can also get an inter-day CV where you can look at sample A on each of the 5 days. Now, by taking the sum of the squares, you can get the total CV for the variance of the measurement of any given peptide that you might be interested in. Okay, so we can look at this for some real samples. So this is work that that I did with Deanna Plubel, who's a Ph.D. student at the University of Washington, and she's looking at peptides in CSF. So she's looking at 944 peptides from 268 proteins in her assay. And she actually took the coefficient of variation of each of these peptides and plotted them as a histogram, then drew a line at 20%, and that's what we consider quantitative, is 20% coefficient of variation. And it turns out 71% of her peptides actually had a CV of less than 20%, so could be used for quantification in the range that we were looking at. And about 30% had a high CV. Now, those can still be used for detection, but not necessarily for quantification. All right, now that we've talked about accuracy, stability, selectivity, and precision, let's talk about the last little bit of assay validation. The last thing you need to do to have a quantitative assay is assessing linearity and lower limit of quantification. And as we get into this, I just want to remind everyone that the measurements are all relative. Everything is relative. So, for example, weight. Right? Weight is just relative to this block of metal that weighs 1 kilogram. We just have a 1 kilogram piece, right? And so if we want to measure a narwhal or a kangaroo, that's all relative to this block that's kept in super safe under double-chambered glass. Even our atomic scale, right? It's just relative to carbon. Carbon is 12. Carbon-12 is 12. We just said 12. It's not actually a mass, right? And I want you to keep that in mind when we talk about quantification, because we're always doing relative quantification, and there are always caveats to that. So I'm going to talk briefly about 3 different ways to quantify. We can use an external calibration curve, you can do a sample dilution series, or you can measure differentially, all right? So we'll start with external calibration curves. And so just bringing you back to your Quantitative Analysis 101 class, right? Let's look at a real calibration curve. It's actually a sigmoid. At the very bottom, you have a limit of detection. Your lower limit of quantification is right about that inflection point, and your upper limit of quantification is the upper inflection point. In between these two, there is a linear dynamic range. Some rules of thumb when you're building calibration curves is you want at least 6 points over the course of the calibration curve, specifically 1 at your blank, 5 in the linear range, preferably 1 at your lower limit of quantification. When you are preparing these, what you should do is actually do kind of a hopping with 2 different dilution series. So you prepare your top concentration, then you prepare your second concentration, and you do serial dilutions of each of these 2. And this allows you to see if there are any pipetting errors when you prepare your calibration curve. And we can make calibration curves with either peptides or proteins. There's some pros and cons for each. So peptides are easier to get, but you don't get any of that digestion variability. And people do this a lot. People are really into synthetic peptide calibration curves. A protein calibration curve, however, accounts for all your sample preparation variability, and you probably already have them from developing the method. One other thing I want to comment on about peptide and protein calibration curves is peptide calibration curves cannot be used to tell you the concentration of a protein in a sample. You are doing peptide quantification, not protein quantification. And so really all you're doing is defining a minimum amount of a protein that must be present in the sample. But depending on the digestion efficiency of that protein, there may be more. So I just want to leave you with that. If you take nothing else away from this about calibration curves, it's that peptide calibration curves cannot be used to actually calibrate for a protein. Another nice thing about these protein calibrators is just like in method development, they provide multiple targets. So you can actually plot 5 different linearly responding peptides from a specific protein and pick out which one responds best. In this case, the GLY peptide, assuming it's not interfered with and it's unique in everything, would probably be a very good prospect for use for quantification. We can also use this information to finally do our last little refinements on our transition list. So for example, our normalized peak area here is shown on our right, and 5 different concentration points are shown on the x-axis. And we can see that this blue transition really isn't bringing much to our method, right? It's just kind of floating up our signal and increasing our variability. And if we look at a different peptide, this green transition is really not doing anything. Actually, none of these transitions are doing anything. This peptide is not changing response with concentration. And so this whole peptide can get cut. And these are things that our calibration curves can show us. We want to see this linear decrease of our transitions and our peptides with concentration. We can do this in a label-free or a stable isotope labeled method. So a stable isotope labeled method, you would spike in heavy labeled peptides of interest at a background level and normalize everything to these peptides, okay? Here's what a calibration curve might look like for it. We've got a beautiful R-squared value, nice and linear. This is a beautiful way to do these experiments, great for normalization. You do need at least 95% purity light and heavy peptides, and those can be kind of expensive, right? Now you're buying a lot if you're spending this much on it, but it will last you for a while, but it's a pretty big front-end investment. Now, if you're doing label-free, you're just going to look at the protein you want to see, but you still need to normalize to something. And so these are where those external QC samples, right, the Proteus QC, that sort of thing, is really useful because you can use it to normalize all of your peak areas. And if you do this, you still get really, really nice results. So here is for another peptide, and we're looking at the label-free data normalized to my internal standards. And our R-squared value is also beautiful, right? So all of these work quite well as long as you have very well vetted peptides. Now, you can use this to determine the LOD and the LOQ, right? So there's a MS Stats package that goes into R and is compatible with Skyline, which is a software developed by the MacCoss lab and is freely available. And it's called nonlinear quantlim. And what it does is a bilinear fit of the data. And so I'll show you some of this to show you what that actually looks like. Some other features of interest for MS Stats is that it does integrate with Skyline. It has quant and stats tools and lots of plotting options. So you can do volcano, you can do residual. I actually just found yesterday the TMT quantification is also included in MS Stats if you want to do that. This is what the data looks like when you plot it. So you see your little hockey stick sort of calibration curve here. And let me show you some real calibration curves. Okay, well, this is what you might actually see when you do calibration curves. So here's a big zoom out of a super, super linear one. This one looks great. I didn't even get low enough to hockey stick it. You might see something that hockey sticks really nicely. You have lots of variance in your in the low concentrations. Or you might see something that's basically flat and never gets above the 90% upper bound of noise. So basically is just noise all the time. You picked a bad peptide. But you wouldn't know unless you actually did these experiments, which is why they're so important. And you can use MS Stats to get your LLOQ, okay? And so it actually, when it plots those curves, it just pops it out. Or just writes out the LLOQ for you, as well as the LOD. And I just wanted to leave you guys with a warning on this of just know where you're getting your definitions from, right? So some things are defined by variance, right, based on variance for your LOD and LLOQ. And sometimes your LOD and LLOQ are defined based on a signal-to-noise ratio of the background or the blank. But it's important to know these things. All right, so I'm going to switch gears a little bit here because I've talked a whole lot about spiking something into an exogenous matrix. In this case, most of this was done in chicken serum. But what if I don't have a good matrix, right? What if I only have the third and worst matrix? I have to use my sample itself as a matrix. And so what you can do is called standard addition, all right? So external standard, you would be spiking something into a matrix that does not have any of that compound in it to begin with, whereas with standard addition, you're spiking into a sample that does contain it, okay? So your sample is shown here in black, or would be a response, and then a mocked up sample response, if you spiked it, is just a little bit higher. And this also should be linear, right? That's why we can take advantage of this, is that the linearity should be the same. You can measure your sample and then different concentration points when you spike it in. You should be able to draw a line through these points and be able to determine your limit of detection. Now, you can't assess your limit of quantification you can only assess whether your sample is above the limit of quantification. So you can say, oh yeah, I can quantify this, but you can't say, and I can until this low, right? But this is still important information and useful information. So another way to quantify is actually by doing just a dilution series of your sample in plasma. And so we actually did this for an Alzheimer's disease study that we were working on. Alzheimer's disease is a really huge health crisis right now. It's estimated to have a $1.1 trillion healthcare cost in 2050, and it's known to be based in protein. So looking at apolipoprotein E, there are 3 different variants that are either normal, high risk, or protective for Alzheimer's disease. And so what we actually did here, we're looking for different proteins in Alzheimer's disease, and we just took our human sample and diluted it up in chicken. Okay, so our 100% concentration is our human sample, and our lowest concentration is chicken. And we did do our 2 different dilution series, right, to make sure that we take care of any systematic errors. And what you can see is when you do this, you get nice linear results. So this is for the protein, the peptide IHW from CO3. Okay. You can also use healthy disease pools. So for example, if the chicken serum didn't work, I could use the Alzheimer's disease sample and then a healthy sample and do dilutions of the 2 pools to demonstrate that the measurement is linear between the 2. So for example, I can take 100% healthy down to 100% Alzheimer's. And so if we show this data, say for ApoE4, some mocked up data, what you would see is that it's linear in between the healthy and diseased patients. So with that, I just want to kind of summarize the important things about targeted method development strategies that we've talked about today. So things to avoid are predictive algorithms, data repositories, and DDA data. These don't accurately reflect what you're going to see by SRM. How to make a gold star assay, measure your peptides of interest, evaluate the stability and matrix effect, do all of these evaluations, include peptide and protein standards, and you have to calibrate. You can calibrate in lots of different ways using external calibration curves, standard addition curves, or using healthy disease dilutions, but you have to calibrate to be quantitative. With that, I would like to thank the MacCoss Lab, because again, this work was done in the MacCoss Lab, the NIH for funding, as well as the Genome Sciences Department for funding, and everyone who helped me with this work. I also want to put up some resources for everyone. So this webinar will be posted for 6 months. And so what I wanted to give you are just some resources. So this is a sample preparation protocol for human plasma. So don't worry about writing it down now, but you can come back and pause right here and copy it down if you want to try and use it. I have another protocol here for doing from cell homogenate. Some go-to nanoLC methods for proteomics experiments are here with all of our parameters and the type of packing material and everything we use, as well as some selected resources for further reading on this sort of thing, everything from quantitative proteomics to just clinical chemistry and doing proteomics from there. So with that, I'd be happy to take any questions. Thank you.
Thank you, Dr. Spencer, for your presentation. A quick reminder for our audience on how to submit questions. Simply type them into the drop-down box located on the far left of your presentation window labeled Ask a Question and click on the Send button. Dr. Spencer will answer as many questions as time permits. So the first question is, what is the maximum number of transitions that can be measured at once?
Yeah, so that's actually going to depend on your instrument, and I think one of the biggest things there is how long you have to measure for, right? So your sensitivity is going to be proportional to the amount of time you measure a transition. So I like a minimum transition dwell time of about 10 milliseconds. Obviously, going higher is better, but I found it to be kind of the balance. So with that said, if my dwell time is going to be 10 milliseconds on most triple quads, probably 450 to 500 concurrent transitions you could measure at a single time.
Thank you. The next one is, do you have to do such long digestions, or can you add more trypsin and digest for a shorter period of time?
Yeah, so this is actually a really good point. I didn't talk much about it. One of the slow things about a lot of what I do, and you actually see in the resources I put up there, is that I do an 18-hour digestion for a lot of experiments. So one thing is that a lot of my work was done in plasma, which is notoriously difficult to digest due to protease inhibitors. And so I'm actually using a ton of trypsin in there. But you definitely can add more trypsin, especially if you're using the cheaper, the cheaper grade trypsin, not proteomics grade trypsin. You can add more in. And one thing I'd actually suggest doing if you're going to do that is you add in the trypsin, let it go for 30 minutes to 2 hours, and then do another spike of trypsin.
Thank you. Looks like we have time for one more question. Why do you use both peptides and proteins in your internal standard?
Yeah, so this is a really good question. So both peptides and proteins in the internal standard are because they bring different things to the table. So peptides, you should see, right? So if you don't see your peptide internal standard, there's probably something wrong with your setup or your sample itself. Say you're not picking up enough sample to actually analyze, or your nanoLC tip has gone bad or something like that. But if you can see your peptide internal standard and not your protein internal standard, right, you can specify that, okay, well, the protein internal standard, it should behave like all the other proteins in the sample, right? So you can use that to kind of differentiate where a problem might be, rather than being in the situation where, okay, I spent, hours prepping these samples, 18-hour digestion, whole bunch of pipetting in between, making my methods, getting everything set up. I fire these precious, precious samples that I say got as biopsies, and I don't see any signal. Why not, right? And a lot of the time when you do have an instrument problem, if you don't include a lot of different standards to test for different things, it does turn into, well, I don't have signal, why? And so it's to kind of keep you from being in that position.
Well, I would like to once again thank Dr. Spencer for her presentation. I'd like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today will be addressed by the speaker via the contact information you provided at the time of registration. We would like to thank our educational sponsor, Agilent, for today's educational webcast. This webcast can be viewed on demand through April 2019. LabRoots will alert you via email when it's available for replay, and we encourage you to share that email with your colleagues who may have missed today's live event. Until next time, goodbye.